Yeah, so my name is Dr. Annie Evans. I've actually been with Metabolon for almost 20 years now. So when I first started here, we were just a startup, so all of 12 employees. So I've been honored and blessed to be a part of this fantastic organization for that whole time. Metabolon is a metabolomics profiling company, and we'll talk a little bit more about what metabolomics is. But we've been around since the year 2000, and we're located in Research Triangle Park in North Carolina. Metabolomics, I love to start out with the sort of what is metabolomics by taking it maybe to a term people have heard more often, which is genomics, right? Genomics is the study of genes. So people look at mutations in the gene and try to understand how that mutation impacts the health of an individual. Metabolomics is essentially trying to do the same thing, but rather than studying genes, we are studying the reactants and products, or all of the small molecules that are present in our body that are essentially the outcome of all of those genetic mutations and proteins translations. And what we really love studying about metabolomics is that you can actually capture everything that's happened in sort of that traditional hierarchical, your biology hierarchy, right? So genes get translated to proteins function or not, and you can tell they're functioning or not by whether their reactants and products are changing. So that's what we're testing. That's what we're looking for. And so Metabolon is a company focused on looking for changes in small molecules, either in a very broad view, trying to profile as many molecules as possible, or all the way to really just honing in on a subset of molecules and really diving into specific areas of biology. So that's what we call targeted or untargeted type approaches in metabolomics. But that's what we're studying, and that's what metabolomics is, the study of small molecules. Small molecule analysis actually has a lot of benefits over some of the traditional genetic type basis or even the proteomic basis. Just to speak to some of the challenges, we actually don't know a lot about our genetic code. There are many mutations that people find in the genes, but they're genetics of unknown function. It's a gene of unknown function, or we don't even know if that mutation is what we call penetrant, meaning the mutation actually has an end effect. By studying small molecules, you can see whether those genes are penetrant, whether they actually have an impact on the individual or the being you're testing. But the great thing, and there's always sort of a joke, but just wait, there's more, folks. Not only does metabolomics allow you to capture your own endogenous biological processes, it also allows you to capture nutrition, microbiome impacts. Like in our generation, I love to use the analogy that we have discovered a new organ in our bodies, and that organ is the function of our microbiome. We are just beginning to understand how important the molecules produced by the microbiome are to our health. That is entirely exogenous biology. That is not mapped when you look at an internal human being's genetic code, right? It is an entirely separate set of entities that contribute to our health, but also things like the drugs we take, the molecules we're exposed to, like flame retardants and the dyes in our food. All of those things can be monitored, detected, and actually used to help differentiate individuals from one another, because those things impact our health just as much, if not more, than our genetic code. So that is a really unique and incredible thing about metabolomics, or studying these small molecules, is that better capture of what we are exposed to, in addition to what our own genetics have us pre-wired for, if you will. Some of the most exciting work that we've done is really using both data streams, right? We know that there's a lot of really interesting data about people being, pre-wired for some genetic conditions, but ultimately it is, how does that impact the small molecules? And we've actually done a lot of work where the molecules that we've identified, and we've associated that back to the genes and been able to identify gene function. So there's this beautiful dynamic.
Gene functions, wow.
Yeah, exactly. So there's been a lot of work in that space called GWAS, genome-wide association studies, where by studying the small molecules and knowing the genetic mutations, you can start making those correlations. And in fact, we've identified a lot of gene functions because you study what is the input of a mutation in that gene. So together, they really build on each other in terms of We've collected so much genomic data, metabolomic data is just going to build on our knowledge of that. One of my favorite examples to share with people is I do feel like in the surgence of proteomics and genomics that have happened over the last 20 years, we've almost forgotten about the incredible power of small molecules. When you look at most of our diagnostic tests that we're used to, you go to the doctor, you get your glucose measured for diabetes, small molecule. You get your cholesterol tested to see about what your heart's doing. That's a small molecule. You get creatinine measured to see how your kidneys. All of the sort of diagnostics that we rely on currently in our own medical world are small molecule based. And it does seem like in the last 20 years or so, we've been so excited by the genomics, we've forgotten about the massive underpinning of how much data there is and how much important value is in the small molecules. And particularly now with microbiome stuff coming up and the uniqueness of that, metabolomics is absolutely primed to be able to lead healthcare moving forward. So some of the areas of metabolomics that we're really excited about right now is the ability to move this into more marginalized populations. A lot of my research in recent years have been, can we do metabolomics? Can we do small molecule profiling in dried blood spots? So we had a fantastic study with a collaborator of ours where they were working to understand malnutrition in Mali. And it is a very poor country. And there is some staggering level of like 70 or 80% of children are malnourished and have stunted growth. And so this particular researcher, her name was Elizabeth Ryan, was looking at strategies to be able to look at intervention, nutritional intervention, but they couldn't have cold storage to be able to maintain samples, or much less a phlebotomist or a centrifuge. All of the sort of core things you think about when you're trying to collect a sample, really not possible. So we'd have done a lot of research about collecting dried blood spots, where we actually just poke your finger, take a couple of drops on a card, just stick it in a bag, room temperature, and the data from it is phenomenal. So we're really excited about how powerful this technology is, but also how amenable it is to be able to getting it to places that are much more difficult to get to. Even something like your average veterinary office doesn't necessarily have all the things needed to prep these samples, but if you can just have them collect dried blood spots on a card, you get access to, all the pets as well. And it's amazing how the molecules are so stable that they are robust enough to be able to be analyzed in that sort of a way.
Yeah, wow, that's crazy.
So I'm just excited about, again, if you haven't already noticed, I really profoundly believe that metabolomics and the studying of small molecules are going to revolutionize healthcare. I just use the example of 3 molecules we use right now, glucose, cholesterol, creatinine, to monitor our health. We can study 1,000 molecules. We can detect and identify 1,000 molecules. Your mind almost can't capture the scope of our understanding of human health, when instead of looking at 3 molecules to capture a couple of things, we're going to look at 1,000. And I want to bring that to everybody, because every time I say, isn't it amazing that we can detect how much pesticide you're exposed to in your diet, how many flame retardants you're exposed to in the diet, how well you clear Tylenol from your system every time you take Tylenol. Everybody I talk to is like, I want to know that about myself. And yes, we want you to know that about yourself too. So it's an incredibly exciting time in small molecule research, and we are just enthusiastic about the potential. The nice thing about the Metabolomics conference that we just had is it really highlighted one of the core values of Metabolon, is that we are a client-focused organization. We want our clients to be so successful at what they do. And in fact, everything we do is really about making sure we're good collaborators, making sure that they understand the data, making sure that they are empowered to have success. And so the Metabolomics conference, it just highlighted that. So we had so many of our clients presenting at the Metabolomics conference. Some of the plenaries even were talking about, I just remember getting goosebumps when I heard somebody presenting on our data, looking at mental health, which is one of the categories of the Metabolomics Society. This is a traditionally very difficult to study area of biology. And the fact that people were using our technology to start peeling apart the layers and the complexities of mental health. It was looking specifically at mothers and their impact on children and how the children then develop, and seeing markers that you would expect in sort of mental health, dopamine and serotonin and all these neurotransmitters, that it makes sense that those are the signatures that are coming up. It was just breathtaking to see that. It was also wonderful to see one of our collaborators, we talked about one of the unique things about metabolomics was being able to look at the drugs that we take. And in clinical trials, you're asked to report, which medication did you take using for patient stratification. And I don't think anybody's doing it like for terrible reasons, but people just don't know or don't remember the medicine that they're taking.
Yes.
So oftentimes we'll see people underreporting the medicine that they are taking, and that can have impact on clinical trial outcomes. So some of the research was also about how they've used metabolomics to really understand who is reporting, and again, not for, they might have just forgotten, but who is reporting what medications they are actually on during the study for better patient stratification. And I found it was actually, I don't think so. One of our guest speakers was a wonderful woman named Anya Halima, and she talked about how she was doing some research into this. And one of the things that she had found was that there was a vast underreporting of going back to mental health, medicine people were taking related to mental health. So people drastically underreported medication for depression and for anxiety. And I think there's just some stigma around medications around that. So people are underreporting it. So I think it just speaks to some, societal pressures about what you are comfortable sharing, what you are willing to share, but then also needing to know that information to really get the most impact from data. So again, it was a beautiful, just a wonderful really coming together of the power and that metabolomics brings, and just showing people using that data in really trying to improve clinical outcome data, improve our understanding of nutrition, improve our understanding of metabolomics. So it was wonderful to see the success of so many of the people that have worked with us, because that is our goal. We just want to drive science and make sure everybody is as successful as they can be. So it was a very good conference for that. One of the things we're most excited about Metabolon is that Metabolon has been, I think I mentioned this before, we've been in the industry for over 20 years now. So we've been studying small molecules for 20 years. So we know signatures of inflammation. We know signatures of kidney failure and liver failure. We have all of this incredible knowledge that we've gained over the time of what are the metabolic signatures of a variety of different biological systems. And so what we're really working for into the future is to start really giving a little bit of a more of a nod to some of these biological systems. So maybe everybody doesn't want to see all of the metabolites in one. There's incredible power in that, but maybe you're really just interested in understanding inflammation in your system. Maybe you're just interested in understanding the specific signatures of diabetes. So what we've been really working on is starting to sort of cull out specific disease areas and have offerings related to those things to just help. Again, if you've already got a hypothesis, you don't need to do some sort of discovery all out. You can really hone into some specific disease signatures. So this year we've launched a suite of targeted panels. So again, that's sort of a more focused look at the metabolites, but of different groups of metabolites that have been known to have influence on inflammation. So if you're really trying to cull apart, we're trying to understand the role of inflammation in your particular disease area, whether it be rheumatoid arthritis or even COVID, you can actually start diving into that. And we have the same thing for diabetes, and we're going to be expanding that into the future. But again, it's just, we have so much knowledge that we've gained about what are the metabolites impacted, microbiome, and that we really would like to start helping people. If you've got a more focused look you want to take, we're going to help you with that so that you don't necessarily have to do a full profile for whatever your research study is. So that's something we're really excited to be getting into, because we think there's, a lot of people who don't necessarily want or need the whole list, but they really want to understand inflammation. They really want to understand kidney performance and just get a deep dive into that area. So that's a little bit about what's coming. Yeah.